Okay, cool. So I also have very big steps, or very big shoes to fill. I don't know very many, probably fewer than one mushroom folks, but I'll do my best. So I'll be telling you a little bit about the making lab that I'm setting up at the Francis Crick Institute, which is just about 10 minutes that way across St. Nicholas Station. And as you can see, it's a building site right now. The lab is due to open soon, but there are a few things that need to be figured out first. And so I've actually titled this talk, Making the Making Lab. And I'll tell you a little bit more conceptually about what we're going to be doing and how we aim to change the way that science is done, both for us and folks working around us in this. So the Making Lab is, we try to focus on these several key areas, and we're going to touch on a few of them, not all of them, so I won't bore you. First, a little bit about the Crick. They say that their first strategic priority is to pursue discovery without boundaries. And what I think that means is bridging the physical and life sciences to do things that aren't commonly done in life sciences. But what they have given me a little bit of a hard time with is erecting a wall that borders my lab, and that yellow arrow is pointing to that wall. And believe it or not, that wall had to be shipped from Spain, and the shipment got delayed, and that delayed the lab opening. So despite the fact that the Crick is telling us that they're pursuing discovery without boundaries, they're not giving me the wall or the boundary. Anyway, so I think the really key concept that Making Lab tries to get at is that we want to be imaginative. The complexity of biological problems today and the intricacies oftentimes demands an interdisciplinary approach between life scientists and physical scientists that requires a lot of failure and novel and imaginative solutions. And those things are not easily pursued in traditional labs. And so we try to create a space where people can co-work and collaborate and fail together and succeed together. And we are aiming to foster that spirit through being a few things that I'm going to elaborate on a little bit: bold, open, collaborative, and communicative. So first, we're trying to be bold, and this will give you a little bit of an idea of what we actually do. So first, we focus on microfabrication, so things like photolithography to generate microfluidic devices and micropattern devices. A little bit of macrofabrication, so 3D printing, CNC milling, a little bit of electronic fabrication, PCB, manufacture and developing sensors and actuators, and all of those areas of fabrication overlap and allow us to create some pretty cool devices. We are trying to be accessible, and that means a few things. First, there are some companies that exist to do some of what we do. They have some drawbacks. None of those companies are embedded in the Francis Crick Institute like our lab is. And oftentimes, as most of you know, or all of you know, solutions to scientific problems require multiple steps. And the company that can provide a solution to step 1 is usually not the same company that can provide a solution to step 2. It's not the same company that can provide a solution to step 3, et cetera, et cetera. And that ends up costing you, as researchers, a lot of time and money, and we aim to overcome that. Secondly, the traditional areas where this kind of physical science and life sciences have been done require either a clean room, as indicated in the picture on the left, or in this kind of very stereotypical image of a molecular biologist, unlike what we just saw. And neither of those venues are very conducive to communication or collaboration. And so the physical space of our Making Lab is diagrammed here, and don't worry about reading any of the details, but just suffice it to say that it's a space that contains benches where physical scientists and life scientists can work together in the same area. And as you guys know, the refinements that oftentimes underlie scientific successes are out of reach in the traditional settings, and we're aiming to kind of expedite the process, make it more efficient, and more cost-effective from a cost and time standpoint, and allow people to work together. We're aiming to be collaborative, and that means that first, we don't need that much equipment or resources to make the stuff that we're going to make, and there are a finite number of things that we need to possess, and we aim to teach individual researchers and train them on the pieces of equipment that do exist. And in this cartoon diagram of people, notice that they're arranged horizontally. That was very specific. We aim not to create a vertical environment where there is, say, a director of the lab and then people underneath that director, but more of a horizontal environment where one should feel free to talk to anyone else and share questions, curiosities, failures, successes, etc. Part of our rationale for doing this is that there's a lot of experience and expertise amongst other Crick core research groups that we aim to take advantage of in the first instance. And finally, we're collaborative not just within the Crick or within London, but internationally as well. There's a wonderful new Microfluidics Institute in Paris called IBGG, and we collaborate a lot with them, and I might say that we've hosted at UCL, where I'm currently doing my postdoc, these biological making workshops that have been pretty successful in 2015, 2016, and I know that a few of you came to those. The next edition might be at the Crick, it might be at IBGG. If it's in London, I encourage all of you guys to come. I think it's a really fun time. And finally, we aim to be communicative. I think we all in this audience believe that the impact of any scientific research is limited by our ability to communicate to an external audience, be that an external audience of experts or an external audience of non-experts. And that's part of the reason why I'm really happy to be here talking to you guys today. And luckily, the Francis Crick Institute and other affiliations I have both external and internal mechanisms to disseminate information that we're going to learn. So I hope I've given you a little bit of a sketch about what the Making Lab is going to be. And I might give you one application. And this was a really fun project that we started when I was a postdoc in Dresden, Germany. And Martin, who's the lead author on this paper that came out last year, and we did this funny thing about putting cells in microfluidic channels. So basically we put individual cells in miniature plumbing. And we didn't think much interesting would happen, but we were really surprised when the cells started moving. Like, well, what's going on? And it turns out that when we made the plumbing a little bit bigger, the cells slowed down, and we made it a little bit smaller, the cells sped up. That was a little bit puzzling too. And we tried to come up with explanations. We failed with long explanations, but we finally hypothesized that maybe a cell's ability to move has something to do with the friction that it can exert on its environment. And so we created, designed and manufactured this microfluidic device, which is kind of schematized here, that allowed us to measure the friction that cells exert with their surfaces, with their surrounding environment directly. And the results of those friction measurements were consistent. That is, the higher the friction, the more efficiently the cell can move. And I wish I could have embedded the videos in this presentation. Sadly, I couldn't, but I encourage you, if you're interested, to read the paper and dig into supplementary material a little bit, where the videos actually are. So that's what I wanted to tell you guys today, and I hope that's given you a little bit of a sketch about what the Making Lab is envisioned to be, as well as one example of an application. And by all means, if anything is unclear, feel free to talk to me more in the panel or afterwards, just outside. Thanks.